Um, Thank you, Rafi. I forgot to record it. If everyone is okay with recording, you're going to record the session. No objections? All right. So legally, that's what you have to ask when you start the recording, and it has to be part of the recording. So I learned that recently. All right. So the organization was formed in 1983, and we were registered in 1984. We have been around for 40 years, 41 years, and we are focused on professional development. We have done a lot of projects in Armenia. Part of the projects that in Armenia was basically for us not to own the projects, but fund or help with the funding of the research and development in Armenia. Some of the projects that there were proposed to us, one of them is Vahan's project, that was proposed to us, and it was one of the winners of our R&D fund. In general, the organization has two sections right now, juniors and seniors. The junior section includes the Science Olympiad, the robotics competition team, and the RMAT programming and coding classes. And the seniors department right now, we are starting a faculty affairs committee. We have the mentorship program. We have the yearly, we repeat the conference and conference every 2 years, and then we participate at Glendale Tech Week. We offer lectures, we offer workshops, and so on. So our future events are on July 14th. We have a Vartavar fundraiser picnic planned. It's supposed to be a networking event, but more casual. And we also have a workshop specifically for engineers from Armenia to improve their resumes for positions in US if they want to work remotely with the US. That workshop is on the 25th, so you can check our social platforms and find those information. And by the way, I forgot to introduce myself. My name is Linda Megerdician. Currently, I'm serving as the president of AESA. With that, I'll just invite Rafi to give us a brief explanation about the R&D team, and from after that, I will ask Vahan to continue with his presentation. Thank you, guys.
Thank you, Linda. So my name is Rafi Anfisio. I live in Berlin. I'm a research engineer. I joined the ASA in end of 2020, beginning of 2021 to start developing the R&D program. Apparently, this was something that was in discussion since several years before, and then we started forming a team and discussing how to develop this. So we spent a bit of time in defining the scope of the team, what it can do and what it should not be doing. That took also a bit of time just to start to see the ball rolling. And then we developed the RFP. So the RFP is about scoping what type of research project we want to see happening and funding. And the design of that was to try to include as much as possible the development within Armenia. And structurally, the RFP funds projects where we have people on the ground directly in Armenia. Whether they are or not of Armenian descent is not really something that we wanted to enforce, but since this is in Armenia, then this was mostly the case. We wanted also to have the type of areas we wanted to fund, but here we left the door to open to various type of research as long as they were state-of-the-art and cutting edge. And they were about prospering Armenia in general. So whether this is about the well-being of the people in Armenia or various scientific areas. So this was until 2022, we developed this thing. And then we we started announcing for the call for projects in July 2022, and we collected various projects here until September 2022, October 2022. And in the meantime, we started creating a review panel for all those projects in various areas that we wanted to cover. While we were receiving the various applications, we expanded this review panel to cover the topics more precisely. At the closing of this RFP, it was then in October, we started the review process, and we basically collected the feedbacks from all the reviewers. So basically per project, we had 2 reviews from people that were either expert in the field or expert are senior enough to tell about the project. We also shortlisted various projects based on the requirement that we had in the RFP. And then we selected 3 projects. So the one of Vahan Grigorian today is one of them. And based on the financial support they wanted, we tried to accommodate as much as possible the fundings. We then started funding the project, I think, in February 2023, and we started discussing on how to fund the project. We had a bit of issues in how to deal with the money and or how to send the money there and there, and but apart from that, it was all good. And I think the funding started in July 2023 for 1 year. And now Vahan's project is at the end of the funding, and we released the latest batch of funds recently. And that was more or less it.
Thank you, Rafi. Thank you so much. Vahan, John, can you please? I can hear myself twice, actually. I apologize. Do you guys also hear an echo?
At this moment, no.
Okay, I hear my own echo, but
Now we do.
oh, okay. Let me just try this.
But it's fine.
How about now? Let me just do this. Thank you, Rafi. Okay, let's start. Vahan, would you please give us a brief, basically explain about yourself and continue the same topic that
Sure.
we're on? Thank you.
Sure. So, dear respected experts, dear colleagues and friends, first of all, thank you for having me on board. Today, I will discuss the research project that we are doing in the context of our research group. First of all, please let me introduce myself. My name is Vahan Grigorian. My specialization is orthopedics and traumatology. I'm doing my PhD project currently in the Laboratory of Tissue Engineering and Immunology in Orbeli Institute of Physiology, and also I serve as the head of the Department of Science of the University of Traditional Medicine. In the context of this funding, our team consists of 3 specialists, but we have also others, and I will introduce them in the end of the presentation for sure. So the topic refers to tissue engineering of peripheral nerve tissue. Because our audience consists of specialists from different backgrounds, I will give a little bit more introduction. So first of all, when we talk about nerve tissue defects, first of all, we need to discuss the influence of different traumas, and these traumas can be high energy or low energy when we have loss of nerve tissue. But also in case of different surgical extensive tissue resections, it can be due to cancer or it can be due to any large-scale lesion. This is typically a microsurgical intervention, of course. In this case, when we have extensive loss of tissue, we can also have at the same time nerve tissue loss and defects. So this is the classification for nerve injuries. Sorry. So neuropraxia. This is the mildest type of nerve injury. In this case, we have only physiological injury, but we do not have discontinuation of the tissue itself. Of course, this image is quite schematic. So this yellow line represents the axon, and the red lines, as here is seen, represent the surrounding layers. So in case of axonotmesis, we will have a rupture or injury to the axons, but the nerve itself will still maintain its continuity. So we do not have a rupture of a nerve per se. And we have a neurotmesis, which is already a rupture, which can be partial rupture or complete. In this case, the surrounding tissues inside the nerve are also damaged. So after the injury to the nerve, a process which is called Wallerian degeneration starts, and it goes both in anterograde and retrograde way. So in retrograde way, of course, we have some changes into the cells, cell bodies. First of all, there will be swelling of the nucleus and other changes. But also this Wallerian degeneration specifically goes a little bit also backwards. So this is retrograde. It goes until one node of Ranvier, which are the structural components of the nerve. But a distal part, which already goes and innervates the end organs. In this distal part, it's gonna be completely degraded. So the major cells which play a role in this process are the Schwann cells and also the macrophages, which infiltrate to this zone, and then they get rid of the debris of the axon distally from the injury side in order to free the way for the proximal from the stump for the stump to regrow throughout these freed tunnels. Let's put it this way. And here we can see on the right side of the slide the extensions that the axonal stump forms. Major extensions are called lamellopodia and filopodia, and by these extensions, it can, let's put it this way, like coordinate around its surroundings. And if the regenerative tunnel is intact and free of any obstacle, it will gradually regrow and reach eventually the end organ. Of course, here are special conditions that are needed to be met in order for this to take place properly. So first of all, when we talk about this regenerative tunnels, in this case, we need to make sure that the continuity is present. If we have a complete or even partial rupture, this is becoming quite challenging. Of course, in microsurgery, there are techniques for end-to-end suture. You can suture it, but of course, with proper alignment. However, if we have loss of the tissue, aka the defect, then already there is a need to weigh down all the pros and cons of each specific approach. So conventionally, it is considered that if this gap, if this defect is longer than 5 millimeters, it is already needed to start thinking about nerve grafting. Of course, if we have experienced, plastic surgeons, experts, they can sometimes even larger gaps repair by end-to-end suturing, because it is possible to do extensive mobilization of the surrounding tissues. But first of all, it is traumatic, and then it's not always possible. So we say that from 5 millimeter and beyond, we should already consider the need of grafting. So this is the nerve guidance channel, which serves as the means of restoring this regenerative tunnel. So let's first of all discuss the conventional and golden standard approaches, which have, of course, made tremendous impact in medicine. Of course, the golden standard is the autografting. So in this case, it is possible to sacrifice another nerve from the patient himself or herself, and using this nerve, repair the damaged one, which is functionally more important. Of course, there are some limitations, of course, also advantages. Regarding the limitations, which are more of concern, is that sometimes here, as in the lower left part, you can see sometimes there might be a graft mismatch. So the size of the graft taken might be much smaller than the nerve to be repaired. And in this case, you need to take several parts and then combine them, etc. Then you do or also some extra injury, extra surgical intervention with its all possible complications. You can have end neuroma, you can have functional deficits, even if you are using not so important nerve, extra traumatization. And eventually, the success rate, even for this golden standard, is about 40 to 50%. So we can say that half of the cases is still going to fail, which is quite a large concern. There are some others, again, very interesting approaches devised by microsurgeons, and these are also work. For example, use of venous grafts instead of nerve grafts. In certain cases, the vein can serve as some sort of a regenerative tunnel and guide these nerve axons. However, the vein itself lacks this internal structure, which is isn't similar to a nerve. In this case, there is another technique which uses muscle in a vein, because you know this muscle has this striated structure. It can serve as some guidance, but again, these approaches are even less effective than the golden standard. So another approach, allografting or xenografting, so cadaveric tissue or donor tissue or animal tissue. In this case, of course, the major concern is the immunosuppression, because at least 18 months you will need to do immunosuppression in case of larger grafts for much longer. Which, of course, let's consider we have a patient with multiple injuries, polytrauma, and you do allografting and then you need to give immunosuppression. But in case of large-scale injuries and polytraumas, especially contaminated ones, immunosuppression can even sometimes, unfortunately, mean a death sentence. So this is, if there is a need to restore a vital organ, then immunosuppression 100% is the choice. But for mere restoration of a nerve, this will not be an adequate solution. So then the scientific tendency started to move towards tissue engineered grafts. The pioneers were making prosthesis of nerves using silicone and other neutral materials. Back in this time, it gave some good results. However, these prostheses can cause chronic inflammation. They also need to be removed eventually. Again, extra surgery, etc. So currently, tissue engineering tendencies, of course, go towards biomimetic nerve guidance conduits with all the inherent properties in order to create the favorable conditions for the nerve to restore. So here you can see certain different examples developed by different scientists. For example, here you can see Hitozan-based constructions, PLGA, PCL, which is again widely used in tissue engineering. Here you can see collagen, etc. So here we need to consider that every property of this prospective graft will affect eventually not only the structural aspects, but also the cellular viability and cellular activity. Let's put it this way. Not only considering the regeneration of the axon itself, but also the Schwann cells, which aid this regeneration. Yes, they create this growth cone, and all the surrounding tissues and cells, such as, for example, fibroblasts, which can, if we delay this regeneration process, fibroblasts can create scarring and render the regeneration impossible. So all these aspects need to be considered. Our laboratory and specifically our research project is focusing on the techniques of decellularization, which means cleansing of donor or animal tissues of cells, leaving only the extracellular matrix, and then adding certain additives which could potentially support this regeneration process. This has two major, or we can say three major advantages. First of all, when we clean and get rid of the donor cells, we decrease the level of immunogenicity sexually tremendously, because mostly it is about the cellular component which creates immunogenicity. However, there are some other aspects that we will also discuss. On the other hand, maintained native structure, which is quite important, the micro and macro architecture, especially as you have seen for the nerves, to have the proper growth canals, these regenerative tunnels. And also, there is one quite important advantage, which is also utilized, the bioactive molecules, which are inherently present in the extracellular matrix. And if the decellularization is done properly, upon the natural biodegradation of this extracellular matrix, these inherent bioactive molecules can also be released and further foster the regeneration process. Sorry. Yeah. However, of course, this direction is developing, but there are a lot of issues which haven't been addressed fully. That is the reason why tissue engineering is still not creating grafts which could be better than the golden standard. So this is our aim basically, yes, because the golden standard approach is present. So these are the important aspects that nerve regeneration techniques need to solve. First of all, scarring-related risks, as I said, because as we discussed, if the regeneration is delayed and if fibroblasts are infiltrated in the distal part of the nerve, this scarring can render regeneration impossible. There are in tissue engineering also certain issues whether or not to add the cellular component. This is also important because do we need a graft which has some cells that could support, or we can use acellular ones? There are different approaches. Currently, we are using the acellular options, but there are also options for cellular component addition, and this is going to be our next step to evaluate whether or not this cell use is beneficial in this context. It will also, of course, depend on the extent of the injury, right? Another important topic, which was the main scope of our present research project, is that it is still not fully known. I'm sorry, not fully known whether or not use of electrically conductive or electroactive materials will be beneficial for these grafts, because there is certain experimental data in literature, but it is not unfortunately conclusive yet. So this was one of the major aims for our present research project, but let me also mention that this is a part of a bigger project. So here in this slide, you can see different electroactive materials that are often utilized experimentally for fostering this regeneration, improving the pro-regenerative quality of these grafts, which are called also scaffolds in context of tissue engineering. I will not go into much detail for this slide, but we can discuss in the Q&A if you wish. So currently, we are focusing on graphene derivatives. Initially, graphene was used as an electroactive material for addition in nerve regeneration, however, in nerve scaffolds. However, current scientific tendencies go more towards the graphene derivatives and not pure graphene, towards graphene oxide and reduced graphene oxide. Why? Because they have been proven to be less toxic for the cells. They are more, safer, as safer alternatives, because graphene, due to its specific structure, has a little bit toxic and genotoxic effects. That's why the tendencies go towards graphene derivatives. Here you can see part of our experimental workflow. We are utilizing mostly red models. Here you can see our colleague Anjan Kumar, a team member, working under a stereoscope in order to isolate this nerve. However, this was one of our trial attempts, because the major attempts were done under a biosafety hood, so level 2 biosafety cabinet, in order to avoid any contamination. So here you can see in the figure A the sciatic nerve itself. It is quite small, as here you can see in the measurement. It ranges from 1 to 1.5 centimeter, roughly. This is a widely utilized nerve for the rats, and due to its size, it's quite, how to say, convenient to work with, as compared to other smaller nerves for rat. So we have devised some technology in order to decellularize by combining different technologies that are present in the literature. So there was a technology which was using technique, not technology, technique which was using freezing thawing cycles in order to destroy the cell membranes. We start from this technique, but then in addition we add washes with detergents in order to get rid of this cellular component which has been previously destroyed by these freezing thawing techniques. So as here you can see, the main detergents which are used are SDS, which is sodium dodecyl sulfate, which is widely used in tissue engineering, or SDC, sodium deoxycholate, these two alternatively, combined with Triton X100. And by this consecutive detergent washes, we make sure to remove most, if not all, the cellular component. Afterwards, I will show the histology images which show the removal. Here we can see this image is not that much informative, but this one is stereoscopical imaging. It's like a stronger magnifier glass. It's not a microscope per se, but something in between. Here you can see just why I included this image, because left side you can see before and right side after. Here, these interesting structures which are called vasa nervorum, the small vessels which are present and perfuse the nerves. Here you can see by a red arrow, and of course, because they are cleaned of blood and of cells, on the right side it's not visible. It's not that much informative in this context, but it's interesting to see also it's kind of a macro structure, before we go towards the microstructure. Here you can see this image is taken from histology atlas, the standard appearance of red sciatic nerve. Here you can see these bluish dots, which are the cell nuclei. This is classical hematoxylin eosin staining, and hematoxylin stains a bluish purple color the acidic component, and specifically the nuclei, where we have the DNA, etc. And the cytoplasm and the extracellular matrix partially are stained with eosin in a pinkish color. So here you can see after the decellularization, this is already our own samples. And here you can see that there are very few blue dots which represent the cell nuclei, but most of them are cleansed. Of course, we can see that there is a slight structural disorganization as compared to the classical appearance, but this remaining structure is sufficient for the guidance properties to be present. Here you can see this one was confocal microscopy. We were using the dye called DIO, which can be used both for cell and extracellular matrix staining. Here we stained the extracellular matrix. Actually, it is difficult to show in this picture, but it allowed us to have the 3D structure reconstruction. And by this, but in this image, you can see the cross section. By this, we could also see characterize the structure itself. Yeah, this is another sample, a larger nerve. Here you can see the process of the cell isolation, because we tested these samples in vitro, not in animals yet. We tested them on cells, and this is the dorsal root ganglion isolation, which allows us to isolate afterwards the nerve cells, neurons and glial cells from this dorsal root ganglia. On the left side, you can see this is a rat spine cut sagittally in half, and the spinal cord is removed. We used here a technique described in Jovi journal, which is quite a famous journal of visualized experiments. So according to this technique, we cut in half, we remove the spinal cord. Here you can see the canal, and you can see these pale whitish spots, which are the dorsal root ganglia, and it takes quite carefulness, but it was possible to remove with special tweezers this ganglia. And on the right side, you can see the ganglion itself after the removal under a stereoscopic imaging, stereo microscope. These are the cell cultures that we have isolated from them. So morphologically, these brightish cells are the neurons, and the others, which are marked by arrowhead, are the glial cells, according to this protocol. Here you can see the incubation techniques. Basically, we used, first of all, we tested this nutrition media incubated with these nerve samples, with or without addition of the graphene derivatives. In this case, it was graphene oxide using different detergents. Here we can see this is, of course, a preliminary data. This is not conclusive yet. I would just like to warn in advance. But here you can see that most of the cases using the low concentration of graphene oxide combined with the detergents did not affect the viability of the cells itself. But currently we are repeatedly doing this experiment to have statistical validity. But SDS with a higher graphene oxide concentration was harming to the cells. This is the overall project framework. So here you can see that we are also preparing a systematic review article at this time, but also we plan to, by finalizing the experiments, we will also write experimental article. Then, well, this is a part of my PhD project. This research project is a part of my PhD project, and here you can see those with the dotted lines are prospective future directions. Those with the solid lines are the upcoming activities and some activities that are already done. So we plan, after testing different electroactive materials, to test also different biological, bioactive materials. So currently we were having meetings. I was having meetings with different experts in order to obtain also these biologically active materials. Which are the future perspectives for the project? So, of course, in this stage, we are doing only in vitro on cells testing, but of course, the next and most important step will be in vivo testing in order to compare the outcomes already in the animals. Upon choosing the best combination, then we will try the next step to develop branched grafts. And in case of success of development of branch grafts, we will already think about nerve electronic sensors also as a possible ramification, but it is, of course, at this moment a bit early to talk about it. This is our team. So on the upper left side, you can see the three research group members who are in this specific grant project. It is myself. We have Seda Gasparyan, who is from Orbeli Institute. She is a biochemist, medical biochemist, PhD students, and a junior researcher. We have Anjan Kumar that I introduced and is present in this meeting. He is a student of the University of Traditional Medicine of Armenia, because our research group has been formed as a sort of collaboration between specialists from Orbeli Institute and University of Traditional Medicine. Here we, on the right side, have Goharik Sevan, who is not included in this specific grant project, but has been included in different projects related to this one. So she is also our team member. And we have the supervisor of our research group, Professor Zarovi Karabekyan, who is the head of our laboratory. And she is also an adjunct assistant professor at the George Washington University. She is the scientific supervisor. Regarding the project funding, we have received, of course, ASA funding, but also we had last year in this specific project faculty support grant from Enterprise Incubator Foundation, funded also by PMI Science, and we have this is my specifically PhD support grant from the Committee of Science. And the collaborations, we have this is a center in Russia, in Yakutsk, Graphene Nanotechnologies Research Center, who kindly provided us with graphene derivatives. Of course, University of Traditional Medicine, the Folding Bioprinting Lab. I am a co-founder of this lab, by the way. And we have, as a collaborator, the Kendall Synchrotron Research Institute, which helped us with two-photon microscopy. And eventually, just a little bit about the research, the reporting and dissemination that we have done so far in the context of this project. So we had webinar organized by the EIF and PMI. Anjan Kumar has presented this project in a student conference in UTMA. We have presented also several times different posters. Specifically in this image, you can see we presented the poster presentation and it won third place in the international conference in Orbeli Institute. On the right side, you can see a small abstract published in Russian Academy of Sciences conference proceedings. And on the lower right part, I was presenting this project also this year in Moscow. And eventually, we also organized this year. This was the first one. We organized the TEDx in UTMA, and we invited our scientific supervisor, who also was talking about our project. And on the right side, you can see our latest presentation, which was in YSMU during the neuroscience conference. So thank you very much for your time and for your attention. Feel free to ask questions. I will be glad to answer.
Are there any questions?
May I have a question?
Yes, please.
Actually, it's I will divide it to the questions and comments. It will be in a different sessions. So first of all, congratulations, very interesting presentation and information and a work which your group is doing. As you might know, a group of plastic surgeons in the past, they did a big research about the peripheral nerve regeneration, and actually one of the work was done in a physiology institute using different molecules, including PRP, proline-rich peptides, to stimulate a nerve regeneration. At some point, we stopped this experiment. It was an in vivo experiment on sciatic nerves on the rats. We have some results, but I'm very surprised that you are not using the techniques for visualization of the nerve regeneration developed by the same institution in a lab, maybe 2-3 floor up from your lab where you are located, because I don't remember the professor. Professor Galman was a PRP researcher, but we have somebody working with us.
I'm sorry. Are you talking about the group led by Dr. David Abrahamyan?
Actually, it was led by me, Dr. Gevorgyan.
I'm sorry.
Dr. David Abrahamyan was doing research, and he defends his thesis at that time. When we finished this work, I was already PhD, and I supervised the whole work. So there is a lot of new techniques now for morphology for nerve regeneration. And if you are trying to use these techniques, you will have a better result to see how the nerve is growing. Coming back to conduits, all the conduits, of course, we are thinking about coming with some super conduit which will, first of all, vascularized as soon as possible, because anything which you are putting in vivo need to be vascularized to be survive because if you don't have that vascularization, it will be a foreign body in your body. You need to something which will be revascularized as soon as possible. So that's why many researchers thinking about developing something beside having a conduit, like thinking a bioengineer have some scaffold for helping nerve growth. They are thinking about putting in this scaffold any different type of molecules or a growing factor which help a nerve to grow. And by my opinion, instead of going and taking a culture of the nerve ganglia from a spinal cord, you might think about using multipotent stem cells because they will become cells which help a nerve to grow and develop myelin and help axon to grow through that scaffold. And the third thing, talking about electricity, I think it's a lot of studies showing that always nerve grows very well when it's stimulated by the electricity, and having some kind of scaffold with the implantable electrodes which will stimulate that scaffold will eventually bring a better result. There is a lot of things which can be done.
Sorry, bad results? Or
good
better?
results. Because there's a studies which they are using implantable electrodes which will be dissolved in 6 months, but these electrodes will be there, and through that electrode you are giving electric stimulation, even to the normal sutured nerve without any allo or autografts. And this is very important to have some kind of conduits which will be able to take this electricity from outside sources or through the electrodes. There is some thoughts. Of course, you cannot be able to cover everything in your PhD. There is a lot of things need to be done. I don't know who is your supervisors, or you saw the group of the supervisors, but I think you need to have somebody who did the research in a peripheral nerve regeneration, especially from a plastic surgery department, the people who already did this in the past and have experience working on animals. And the most important thing at the end of your work, you need to show how the outcomes. You need to measure the outcomes. All this morphology is something, but at the end of the day, if you will not show that this step test for rats is working, or you will not show that electroconduit EMG or EKG will show improvement, all your visualization under the microscope will prove the functional outcome. There's a big things need to be done in your research, and I think that you need to find a good people who will help you to navigate through that world. I understand that the people who, when you're working, they have a big experience in bioengineering, but I think you need some good advisors for your research project. Thank you.
Yes.
My thoughts.
Thank you, Doctor, very much for your comments. Of course, this is a little bit peculiar because my background is traumatology, and my scientific supervisor background is immunology and tissue engineer. For me, this topic, when I was a couple of years ago, when I was choosing the PhD topic, was quite new, to be frank. But we chose this topic because of its importance and urgency. And that's why I'm doing my best to establish connections and collaborations with the top experts to ask them for consultations. So it will be great if you don't mind to connect also with you afterwards. And I will be very honored if you could share your insights, because it will be too much valuable for us and for me personally.
Absolutely. I will be happy to do that. And also we have good skilled microsurgeons who were doing our experiment in the past, who are suturing the nerves and conduits under the microscope because it's their skills. And some of I recognize one name on this call. I think Arthur Harutyunyan, he was one of our co-researchers. Arthur Harutyunyan, dues bani bira, katsi bira. iPhone bira.
Yes, of course.
Arthur na. So there's a people who are interested in these topics. They are in this. So you need to choose also the people who can do in vivo experiments to use your conduit and do the microsuturing. It's a technical part of this microsuturing. It's one of the important part because if it will be some technical problems with the sutures, you will never get more results. I will put with some people who can help you with the microsurgery part, and happy to talk with you after this call to navigate you where to go, in which part to go.
Thank you, Georg, for offering your help. Yeah, that would be a good idea to take the conversation offline because I don't know if there are other questions here, but it's really great to know that we have specialized people in the area who can support this project. And thank you, Wahan, for being open to getting the help from everyone, and Georg for offering your help. Actually, it's very very nice to see this. That's why ASA exists, because we are here to create that platform where people can connect with each other and collaborate. So it makes me happy to see that everyone is so open to working with each other. It's great. Thank you, both of you.
Thank you so much.
Are there any other questions among the audience? I have questions, but I think, Valentin, I'll reach out to you separately, and also, Georg, I'll talk to you separately because it's my own personal condition, so I don't want to broadcast it here.
Feel free.
Thank you. No other questions? Okay, so the recording will be published on our YouTube channel, and we will also send an email out with a summary of the project that Vahan has worked on, and well done. It's a very interesting project. I think I was fascinated by the topic, So thank you so much, Van. And Uzu Gorovtazi, success to you. And I hope that you guys make this, capitalize on this, and make this it becomes project that can become a business in future or something because that's the goal for Armenia. That's part of the reason that we're supporting this project, so it can become something very successful in future in Armenia to also help the economy and everything. So I hope it gets there one day.
Yeah, and using this occasion, I would like to also once more thank the ASA, not just because of funding, but also for dissemination, because you have shared our idea with a lot of experts, and also for creating such wonderful platforms which help us to establish important collaborations, hopefully.
Thank you, Vahanjan. Thank you so much for your time, and everyone else who was on the call. Thank you for joining, and look out for our other emails for future. Sarkis, is that Sarkis Bakodaryan messaging? I just saw a message from, yes, I'm pretty sure that's him because he has been working on this topic, neurostimulation for treatment of migraine and etc. So that's the project that he's interested in. If anyone has any comments on that, please reach out to me. I can put you in touch with Sarkis. And our email address to communicate with us is contact@asa.org. Thank you so much, everyone, for joining, and have a great day. This is Hayastan Barigishter, and here have a blessed Sunday. Thank you. And in Germany also, Barigishter Rafijan, almost Gisher. Yeah. Thank you. Bye-bye. Bye, everyone. Thank you for joining. Bye.